that. So I'd like us to go to the next presenter, E.Tovela from Mozambique. Welcome. Presentation in 10 minutes and then 5 minutes of question.
I'm sharing my screen now.
Okay.
Can you all see?
Yes. Maybe put it in presentation mode.
Ah, okay, sorry. Is it okay now?
Not yet. Not
Okay.
yet in presentation mode.
What about now?
But to save on time, if you can still,
Okay.
I don't know if everyone is able to see as you go on.
Okay. Morning, everyone. I'm E.Tovela from the University Eduardo Mondlane in Mozambique. PhD student at Eduardo Mondlane, doing fish taxonomy and genetics for this complex gene, but the Lethrinus. This is part of the project. It's an ongoing project. So now I'm presenting the first results that we got from the work. This is the structure of my presentation. Going to the introductions, like this is the most is considered one of the most difficult genus to study because of the conservative and they are constant conservative in morphological and anatomical characteristics. We have this genus distributed along the Mozambique coast. Is part of one of the big coral reefs biodiversity, where we have at least 19 tree families and 794 species that are already described in Mozambique. This family contains one subfamily, 5 genera, that comprise 45 species, and this specific genus, the Lethrinus, contains the largest number inside the entire family. For Mozambique, it's estimated that we have 14 species that are described that in the fish guide that we are using currently. However, we have not so recent papers, but a study that was done in the north of Mozambique at Vanizi in Cabo Delgado, that shows that that they found 16 species only for that specific regions, and we assume that probably we have 17 or more species in the entire country. The main objectives of this study was to do the identification based on DNA. To provide an accurate fish differentiation within the genus, and to start a reference database for molecular studies in the genus. One of the premises of that DNA barcoding is that a short standardized sequence can distinguish individuals of a species because genetic variations between species exceed that we do. We can, for example, through taxonomy, it's one of the most old tools used to identify species. However, if we are facing some difficulties nowadays.
Excuse me, your slides are not working.
Your slides are stuck.
Okay. What about now?
Yeah, they're still not moving. That's why it would have been advisable if it was in presentation mode.
Do you have more than one screen, Erica? This might be the issue.
Okay. Let me try to use another screen. Can you see now?
No, still the same slide.
Okay, which one?
The first one, the title slide.
Let me stop and do it again. Is it possible to see something now?
Can you change it to presentation mode? It should be an icon at the bottom of your screen. It should be bottom right.
I did it. I'm in the presentation mode now.
On our side, it's still in the PowerPoint.
Okay.
Is there any IT person near you there? Because it's normally a challenge sometimes.
Maybe we can share it for you if that helps. Okay. Or
please. You can see this one?
Yes.
Yes,
okay.
I can see it.
What about now?
You can still see it in PowerPoint form.
What about?
Erica, we can share it for you.
Okay.
We have a copy of your presentation. So Winnie, are you able to do that? Otherwise I can do it directly.
Okay. Please.
Winnie, will you do that?
Okay, let me stop sharing this one. Okay.
Shannon, I only have Esther's. I don't have Erica's. So you can share Erica's if you have them.
Cool.
Okay.
There we go. Can you see?
Yes.
Great. Okay.
Yes.
Okay. Should I start or move on?
Just let me know. You can carry on. Let me know which side you're on.
You can go. Go, please. Yes, you can continue going. Okay, I was here. No, back, please. Okay, I was saying that one of the most efficient region to identify a species is the CO1, because this region has a good power on most of animal groups for discriminations. And this species can be identified from the tiny amount of tissue from juvenile or any material for morphological identification if we face any problem. Next one. Okay, these groups are divided in three, this genus are divided into three groups, where we have species that are considered low-bodied with conical lateral teeth. The other one is high-bodied species with molariform, that is called by Carpenter, that intermediate group, and we have the high-bodied with conical teeth. So basically, one of the most important feature that differentiate this group are the teeth and the type of body. Please move on. So these are the groups that we have species for all different bodies and dental types. Yes, next. One of the problems that we are facing in Mozambique, that it's really difficult to identify these species. As I said, we are in our fish guide, we have 14. However, recent studies shows us that we probably have more, but one of the struggles we are facing is that it's difficult to discriminate one species from another one. And because we have this long coast where every place they give like a local name, and sometimes we don't have like a very precise technique to identify this group, we are facing these problems, and that's why we are pretending to use DNA barcoding to just help us to support our morphological identification. Another problem that we faced was like it's difficult to identify more than 2 species using only a set of primers, where we have to follow another methodology that say that okay, the best way to identify more than 1 species in these groups is to use a cocktail of primer where you can at least more than 4 species identification. Next. So probably this is the list that we have here. We have, for example, the first one, we still have in our guideline, where is red, we still identify the species as sanguineous, but we know that sanguineous is the synonym of Lethrinus massena. We have, for example, Lethrinus elongatus, that is the synonym of Lethrinus microdon. We have only these red ones, all the names are not valid anymore. So probably we are saying that we have, for example, the red ones and the black ones, while we are identifying the same species. So probably this one can lead us to having more or less species identified through this group. Another one is that we have these 3, the last 3 ones. The Borbonicus is not described for Mozambique. However, in our analysis, we found that we have these ones. The miniatus, we didn't found any of them, but we have 1 species from our natural history collection saying that we have miniatus here. And the Lethrinus SP, we didn't know that we could have this one, so that's why we include here in our problems. That is something that we probably have here in Mozambique. Next one. Another issue is the difference between the climate. We are in a very big coast where we have 2 difference in terms of climate influential. So we have in the north, we have influence of monsoons, while in the south we have influence of cold fronts. So because of these specific areas where we are doing our studies, some, for example, in Nyaka, Bazarutu, and Nyaka Bazarutu are from the south under influence of cold fronts, and Ibu, Mekuf, and Memba are in the north under influence of monsoons. Probably we can have 2 different things that we are in somehow identifying them as the same while we are dealing with different species. Next one, please. So what we did was collecting a fin of the fish, doing the extraction, doing the PCR, and afterwards doing the sequencing to have all the species barcoded and identified through the molecular analysis. Next one. Our objective was to study the variations of these species from the taking into account that we have these 2 big regions like the south and the north where we can find this genus. And we want to try to compare the species from one side and the species from the other side. It's like I'm saying that we want to compare north species and south species through barcode. Yes, next one. This is our study area. As I was saying, this is the north area where we have Ibu, Mekuf, and Memba is under influence of monsoons, and in the south we have Bazarutu and Nyaka that are under influence of cold fronts. Next. This is a very complicated tree because we had to include a lot of sequence from GenBank, but I don't know if it's quite visible, but we have like different colors where we have different species based on location. What we are analyzing now in our preliminary results is what we have regarding to the distributions and what we have regarding to the species. It's quite difficult to understand what we have, but next slide, please. So we have these ones that, oh, sorry, it's not possible to see the labels here, but each of these colors represent a species. So, and these are the places where we did the sampling. If you try to see, for example, here, where we are in the Nyasoro, this is one Lethrinus SP. It's one of the unknown species from Mozambique, and we have these ones that is Lethrinus borbonicus that we didn't even know that we have here, but probably we have in a lot of different places. So the results are conducting us for more than what we expect. It's like we didn't expect to have more than one place for this, for example, the Lethrinus borbonicus. But it seems that we have, and we have to go again, if possible, try to collect and try to do the morphology, because we have a lot of species identified, probably has Lethrinus macena, while Lethrinus borbonicus, for instance. And this is the species distribution, where we could see that we have more species in Ibu compared to the other parts. Next slide, please. So this is the example of, for example, these two species that depending on who is collecting and who is identifying, sometimes they can be identified as different ones. And we have these two. This one is from Iñaka, the blue one, Macena, and this the left side is the Lethrinus macena from Ibu. So if someone is trying to identify them using only morphology, could be difficult. This is the small example that I'm bringing here, because we have another ones that are more difficult to identify if we try to compare to the other species. Next one. So because this is an ongoing study, we are one thing that we want to do is to perform morphology. Now we want to try to see based on morphology using a software for the shape, where we can try to see if, for example, one species that we found in the north, for instance, is the same one that we found in Iñaka. Like we have two different sites, but we have the same species that we are considering that is the same wild. Probably we have some small difference on morphology. We want to compare this data with the genetics ones, and we want to try to see in detail this Lethrinus borbonicus species that we found more in Iñaka, but it seems that we have more in the north. Unfortunately, we cannot go more to the north because of the problems that we have now to collect more species, to try to have more things to analyze regarding to borbonicus. We are creating a key identification for this genus through Delta, where we can give new keys for the fisheries sector to identify these species, not only the only way they are using now. And we are trying to create a database for this genus, because we don't have a lot of molecular data in Mozambique. So that's why this will be another big challenge for us to try to set up this database. Next. And thank you. Sorry for the presentation that didn't come out first of all. Thanks.
Thank you very much, Erica, for that interesting presentation on genetics. I'm sorry, because of the time, we shall not take the questions, but if you have any questions to Erica, please feel free to put them in the chat box below.